To initiate translation, one of the first things that has to happen is that the cell must choose the correct start codon. And so this start codon, it is going to be a series of AUG. That's always the start codon, but there could be numerous AUGs located throughout this sequence. So I've shown here that it's located in pink, but notice that it is not at the very tip of the RNA molecule. So it is on the inside a little bit, and the cell does have to put forth a little bit of effort to figure out where it's at. And once it chooses this start codon, that is going to set the reading frame for all the rest of the codons into place. So there are some additional sequences and ways that the cell does determine what is the proper AUG to get started with. Prokaryotic cells look for a sequence called the Shine-Delgarno sequence. And this is going to be within the RNA molecule itself, and it is going to help the cell to actually figure out where this is at. And so the Shine-Delgarno sequence is going to be located over here somewhere. So the ribosome itself is going to be interacting with this Shine-Delgarno sequence, and that will help it to actually figure out where that start codon is located at. If we're talking about eukaryotic cells have their own sequences that they're looking for, and these are what we call Kozak's rules. And so these are going to help us to figure out, again, where is that first start codon that we want to really begin our whole polypeptide synthesis with. So if we take a look at the ribosome and the first tRNA actually assembling here, so this that we see right here, this is the first tRNA with the first amino acid. And the first amino acid, it is going to be methionine if we're talking about eukaryotic cells. If we're talking about prokaryotic cells, it's going to be formal methionine. But that's the one that I'm showing here just in purple on the end. So that would be the first amino acid. This tRNA is going to match up with this codon because it is going to have an anticodon which is complementary to it, and we'll get some good hydrogen bonding between the anticodon and the codon. And then if we also show the ribosome on here, the ribosome, recall, has two subunits. It has a large and a small, and the RNA is really going to be threaded right through the middle of those two subunits. Okay, so here we can see the ribosome is attached. So we have assembly of the small subunit, the large subunit, and we've also got our first tRNA there on the start codon. And this would then be really initiation of the whole process, and we're ready then for elongation. One thing to point out here is that the ribosome does have 3 active sites, and so if we label those 3 active sites, each one of those active sites can accommodate, if we look right here, it accommodates one codon, and it can accommodate a tRNA as well. So this one is going to be what we call the A site, this will be the P site, and this will be the E site over here. So notice that our very first start codon is going to be located in the P site along with that appropriate tRNA that is bound to it. So for the elongation process, what is going to happen now is that the next codon is going to be recognized by the tRNA that has the matching anticodon. So that's what we see happening right here. We have a tRNA that recognizes the codon that happens to be located right here in this active site. And so that tRNA is actually going to come in and it's going to bind to that, and it is bringing with it the proper amino acid. So here we can see that the new tRNA is bound into the A site with its appropriate amino acid. And then the next thing that's going to happen is that the amino acid that is on the tRNA that happens to be in the P site, that amino acid is going to get transferred to the amino acid that happens to be located. That right there is when the peptide bond is formed. Okay, so we can see the results of that here, where we now have 2 amino acids joined together with a peptide bond in the middle. And as that happened, there would have been a water molecule removed because this is going to be a dehydration reaction. What is going to happen next is that the entire ribosome is going to glide down the mRNA and it's going to shift over by one codon. So this ribosome is going to be moving this direction, and when it does, it will have this next codon located in the A site. So here we see after that has actually happened, So we see that the ribosome has shifted down. We see that the next tRNA has come into the A site, and we have that amino acid chain of 2 amino acids. It is sitting on the tRNA that happens to be located in the middle active site. To point out here, notice that we do have an empty tRNA located in the E site. Well, the E site is the exit site. So that tRNA is then going to leave, and it's going to then get recharged, or it will get another amino acid attached to it. So that's the general process that we have. The ribosome is going to continue to glide down the RNA template, moving one codon into the next active site as it goes along. So the amino acids will move first into the A active site, then into the P, and then into the E, which is where they're actually going to end up leaving. We're going to see that this amino acid chain is going to get longer and longer and longer, and eventually we will end up with a stop codon in that A site. So once the ribosome moves to where it does have a stop codon in the A site, that is going to indicate the end of the translation process. Because what's going to happen is that we're going to have what's called a release factor. And this release factor is going to recognize that, and it's going to actually bind to that stop codon. And once that happens, everything will really disassemble. And so that means that the 2 subunits of the ribosome are going to come apart, the tRNAs are going to come off of the mRNA, and the polypeptide chain is going to be released as well. So that would be the completion of translation.